Our next topic is going to be codominance and incomplete dominance. So in horses, there are several different colors. Here we're going to be talking about bay, buckskin, and cream horses. Bay color is like a brown, it describes it here, with black mane and tail. Cream is a creamy color, clearly mane and tail as well. If these two breed, they can make a buckskin horse. So this is like a cream colored body with a black mane and tail. Super pretty, by the way. We are going to exhibit these traits using our monohybrid Punnett squares. So we have a bay stallion. It tells you up here that CB is used for bay. And a cream mare. So CCR. This looks a little bit different because we're giving these almost like little exponents now, and we're also going to make our Punnett square look a little bit different as well. So let's draw our Punnett square, make it nice and large so you can see inside it well. We're going to fit a lot in there. Now, you can't split these. We have to write them together, but if we had just one, then this box would be empty. So we have to double this. It's going to carry 2 of these bay genes. Same thing for the cream horse. You write it out twice, and then we fill in our Punnett square like we normally do. So you have CB, CCR. CB, CCR. CB, CCR. Okay, these are all the same. They match this buckskin genotype. So you would have 100% buckskin foals. Foals are baby horses. If you were to cross 2 buckskin horses, your results would look a little bit different. Make our Punnett square a bit larger. So you are crossing CB-CR. Remember, you have 1 letter over each box. So this would be your CB and this would be your CCR. So that's 1 horse. We'll add the other over here. CB, CB, CCR. Oh, CB. That one's backwards. It's okay, we can fix it. CB, CCR, CCR, CCR. So we're going to convert them directly into percentages. 1 out of these 4 has the bay genotype. So we have 25% bay, 25% cream, 2 of these can be buckskin. So there's a 50% chance that if 2 buckskin horses were to be bred, 50% chance their offspring would be a buckskin, 25% chance that it would be either bay or cream. This shows a blending of traits, which is incomplete dominance. I'm going to skip over and show you another example on the back. The other idea, the other topic, is codominance. In codominance, you see expression of both traits. So on the front page, we had incomplete. It's not entirely one color or another, it's blended together. But think co means both. Codominance, there will be expression of both of the traits. And I'm going to show you an example. Our letters are going to look a little bit different again this time. We're talking about cattle this time. So white, we're going to use the letter W. Red, we're going to use the letter R. Now, we have to have more than 1 letter when we set up our Punnett squares, so we can go ahead and double these. A roan is a color that is present in cattle and in horses, and it's also called brindle on other animals like dogs. It's a mixture of both of these. So it's going to have one of each allele. Now this says, let's say you have a roan sow, which is a female, and a roan calf. What are the possible phenotypes of the father? You can work backwards using this. So we have a roan sow. We know that we have the phenotype RW on one side, and we're going to determine what the father could have been. So if we have two options, we could say that we have a white male, or roan mother here again. Fill these in like we did. We could also say that we have a red father. Would give us roan options here as well. So you're kind of working backwards to determine what the phenotype of the father could be. Again, because both colors are present, and look this up, look up roan so you can kind of see what I'm talking about. We're seeing expression of both traits, codominance. Incomplete means it's a blend of both colors.